Hello. So this video is going to be on proteins. Proteins are a little lengthy. They are also an organic compound. They are made up of carbon, hydrogen, oxygen, and nitrogen. So with the amoeba sisters, that's the CHON. So we had CHO, CHO, CHON, CHO. We did one CHO, which was carbohydrates. We will get to the second CHO, which is lipids, but we're going to go in here and talk about proteins first. So proteins have nitrogen where carbs did not. They have many, many, many different structures and functions. And I have a whole table of them right here. You can have enzymes. Enzymes help speed up the rate of chemical reactions. Some are hormones, which are chemical messengers like growth hormones. Transport, hemoglobin transports oxygen around. We have contractile, which is your muscle. Your muscle is made up of proteins. Protective, your antigens and antibodies, all those good things, or I should say just antibodies. Um, and instructional support, like your hair and your nails. Storage, where we store certain nutrients and toxins. We know, like, for instance, you do not want to get tetanus. That would be bad, bad, bad. All right. Or even we have the diphtheria toxin. And then we have communication, which has to do with cells being able to communicate with one another. So our basic structure for an amino acid is the amino group here. That's the nitrogen. We have that carboxyl group on the other side, and then here is our what we could kind of say is our central carbon, and then we have the side chain. The side chain is which is what is different. If you look here, you have tyrosine and glutamine, and it's this side group here that's different. That makes the difference between those two amino acids. So there's no element R. R just represents the side chain and where those amino acids are different. Dipeptides are when we have two amino acids joined together. It is by a covalent bond, but with proteins, we tend to refer to this in as a peptide bond because they hold the peptides together. Amino acids used to be referred to as peptide residues. That's kind of where that nomenclature came from. A long chain of amino acids could be called a polypeptide, or, and today we just call it a protein. And here is that picture of those polymers. So you can see here, every time we put an amino acid together, we have to do that condensation reaction. We have to pull out the water in order to get, and we can see it right here, the peptide bonds. Protein shape and function. We have four levels of protein structure: primary, secondary, tertiary, and quaternary. We also are going to talk about what happens if we were to unfold our protein, and that is called denaturation. So here are the levels. I like to talk about better with the picture. So the primary structure is the order in which you put the amino acids together. You see valine, glycine, serine, leucine. So, and the DNA has the instructions specifically say, this is the order you're putting the amino acids together. Now, based on that order and hydrogen bonding between the neighboring amino acids, you can get two different secondary structure. You can get a beta pleated sheet, which to me, I always think about when you crimped your hair or it's just pleated, like a pleated skirt or pleated curtains. Or you can get this alpha helix, which is like a curly Q, a little ringlet. All right. It's not, we'll talk about double helix, which has two strands. This alpha helix only has one strand. And notice all the dashed lines on, that's all your hydrogen bonds. Tertiary structure is commonly referred to as being globular. Don't know where that name comes from, but it's when the protein kind of all folds up on itself, or it kind of looks like the last stage. I said this in class. It looks like the last stage of me when I try to fold a fitted sheet. I start off all determined it's going to look nice, and finally I get frustrated and just kind of wad it all up. Done. All right. So my globular fitted sheet. And then We have our quaternary structure. Not a lot of, not all proteins, I should say. Not all proteins have a quaternary structure. A quaternary structure is when you have multiple polypeptides, like there's one here, here, one here, and one here, that they come together to fit as a functional unit. Okay. Like hemoglobin has four functional units that have to come together to work. The other thing, so we talked about all this folding, but what happens if we unfold it? We denature it. When we denature it, the whole function of that protein is dependent on its structure. It's kind of like a paperclip. You know, paper clips work fine, and then you're sitting there, maybe you're bored listening to this lecture, and you unfold the paper clip. You never can quite fold it back so that it works as well as it did before you unfolded it. And that's kind of what happens with most proteins. Once you unfold it, there's no turning back. We have to just break it down and build another one. So here is what we call a native protein. That means it's in its natural state. When you heat it, notice this is right here a beta pleated sheet. We start to get rid of that. We eventually will start to get rid of the alpha helix, and then we just have our straight chain, our primary sequence left. It can't do its job in this form. And then once it cools down, because we generally use heat, that's why you heat things up to sterilize it, is so that you can denature the proteins inside the bacteria. Okay. Once it cools down, notice it looks nothing like its original structure. Now, for some proteins, we have other proteins, little helper proteins called chaperones. And that chaperone will literally um, take that unfolded denatured protein and put it inside of it. It's like a little capsule, and it gives it the proper environment it needs to refold. Okay, but this again is not all proteins. It's not like when you overcook your eggs over easy, you cannot undo that. Okay. Things that will denature protein, we just talked about temperature. The other is pH. That's how a lot of your sushi, they don't cook it with flames, no heat. They put lemon juice, any kind of citrus juices on there that are very acidic to help make it easier to chew. Different environmental conditions, you know, if it's really, really dry or salty, those can all denature proteins also. There is a test that you can do to identify proteins. It's called the Bio-Rad test. And again, this is really kind of, I like this one. It changes the color from blue to purples, which is kind of pretty. And basically what this particular reagent, the Bio-Rad reagent is doing is it is identifying peptide bonds. And if there's peptide bonds there, it will change color from blue to pink. Pink if there's just a little, and then if there's a lot, it goes purple. So here is our two amino acids, our two peptide residues, and we're going to put them together. Like I said, in order to do that, we have to take out our water, and then you can see the peptide bond. Okay. All right, so that is it for proteins. Hopefully, you understood that. And if not, as always, let me know.